I think what I want to do for you is just answer some questions about, I can't tell you about all the things that are being done for research or all the hottest topics, so I can tell you a little bit about how we do research and why we think about research. First of all, a little bit about, so our biggest priority for Dr. Kluger, myself, Dr. Weiss, the nurses, is to take care of you. That's the #1 priority. But close to that are two other things, which is to find treatments that will improve the outcome for people who don't respond to the current treatments. And the other is to teach the people that are following us how to do this so that there will be people in the future who do our job. So those are our #2 tied priorities. But the first one is to take care of you. But we wouldn't be able to do our job well if we did do research, because the standard of care for all cancers, including melanoma, is still not adequate. We're only going to be satisfied when 100% of people get cured with 0% toxicity, and we're clearly not there yet. So why do we do research? And these are slides that Dr. Kluger showed you yesterday. This is the best treatment we had in 2002 or 2003. This is high-dose interleukin-2, and this is a survival curve. And I just want to show you here that at 5 years, only about 15% of people would have been alive who started out with metastatic melanoma, stage 4 advanced melanoma. And you can see here's another randomized trial. Again, survival rates, even at 2 years, were only about 20% in those days. And at 5 years, probably 5 to 10%. Now, this is the first phase 1 trial of anti-PD1 that we performed here at this institution. This is the most recent data, and this is looking at 5-year overall survival rates for anti-PD1. That's nivolumab or Opdivo that you're familiar with. The trial that we started here in 2008 together with 8 other institutions around the country. And now you can see that the melanoma survival rate at 5 years for just anti-PD1 alone, nivolumab, is somewhere close to 35%. And it's actually better than these because all of these patients that went on this trial had had prior treatment. If you really take people who've not ever seen any prior treatment before with metastatic disease and give them anti-PD1, whether it's Keytruda or now Opdivo, either one of those drugs, nivolumab, pembrolizumab, or their other names, the survival rate is probably north of 40%. So we're doing a lot better than we were doing 10 years ago. And so one of the reasons we do research is to try and keep bringing that survival curve up. Now, this is a recent publication. Dr. Kluger and I were on all these papers together. My name is more senior because she's younger, prettier, and smarter, and so she had to give me something. So this is the phase 1 trial of the combination of nivolumab, Opdivo, together with Yervoy, a trial that we started here back in 2009. And this is now the survival curve with substantial follow-up. And you can see that in that phase 1 trial, which then included Memorial and ultimately 2 other institutions, when we take all those melanoma patients, 94 patients went on that trial. And this was a phase 1 study. This was the first time this combination was ever used in humans. We had to find out what the right doses were. We now take all of those patients and we look at the 5-year survival curve for those melanoma patients, it's north of 50%. And so we really believe that with the combination of ipilimumab and nivolumab, or Yervoy and Opdivo, the other names, that probably close to 45 to 55% of patients can be alive at 5 years. And I can tell you that most of those people that are alive at 5 years don't have any evidence of disease, and it's very unlikely that their disease will ever recur. They may be cured of their disease. And again, this is another example of how clinical research helped to improve the outcome in metastatic melanoma. Now, there are randomized trials. Those were single-arm studies, so sometimes you have to compare one treatment to another. And what we found, and this is a randomized study which we didn't lead, we're led by other people, but came from the work that we did, showing that this is the upper arm here, this is the combination of Yervoy and Opdivo, nivolumab and ipilimumab. It's only a little bit better, actually, than anti-PD1 alone. Not a lot better, a little bit better, but still, we believe that the combination is the best. And now what we try and figure out is who can do really well with just anti-PD1 alone, because it's much less toxic, or who needs both drugs together, which has a lot more toxicity. And that's another area of important research that we're trying to do, because again, we're not only trying to get the best outcome, we're also trying to get the best outcome with the least amount of toxicity. So how do we improve outcome in patients? How do we sort of view this? So the only way to do this is to really try and understand the disease, right? And the only way to understand the disease is to the research, the basic research. And for a long time, this research was led here by Dr. Halaban. We have many other people who are involved with this, Dr. Kluger, Dr. Marcus Bosenberg, other people. We try and understand the biology of the disease, the pathways, the signaling pathways, everything. And that leads us to a target, something we can actually target with a drug, whether that's in the immune system or in the cancer cell itself. That leads to a new drug or new combinations. We usually don't develop those drugs. That eventually goes out to the pharmaceutical companies. And then eventually that comes back. Then we then do clinical trials to test whether that drug actually works or not, what the toxicity is, how to manage that toxicity. And during all of this time, we try and collect tissue, blood, and tumor, even in patients who get standard of care treatment, so that we can then go back, take that tissue, go back and try and understand what the drugs were doing, try and better understand the biology. And that leads back into a circle, which leads us to new targets and new drugs. So, and this is just another way of looking at it. We need to understand not only the cancer cell itself, but how the human immune system reacts to that cancer. We need to understand how the immune system works. And we use that information, we translate that information to do interventions, first in the test tube, then in animal models, and then ultimately bring that to the clinic in order to develop better treatments. And all of that is built on a foundation of basic cell biology. These are not things we do. These are what other scientists do, or basic immune biology, things that are basic to how the immune system works. And we borrow from those concepts and apply them to cancer to figure out how the cancer works and how the immune system interacts with cancer. And translation is taking that information, applying it to human tissues, and then applying it to humans in order to improve outcomes. So why do we collect human tissue? Why is it that when some of you come to the clinic, we ask for your blood or we ask for a piece of the tumor? Why is it so important? Well, first of all, we need to understand, not only are we learning from that tissue, but when we do studies in the mouse, we need to know that the studies that we do in the mouse are translated into the human. They're valid in the human. So we have to figure out whether the mouse biology is the same as the human biology. Is it target expressed? Does it have the same function? Sometimes we need to figure out whether the drug concentrations that we're giving to a human match what we gave to a mouse, because sometimes if we don't get the same drug levels in a human, we won't see an effect like we're supposed to see, like we've seen in the mouse. Sometimes we're trying to figure out whether the immune response, which kills a tumor in a mouse, whether we get that same immune response in the human, because if not, then we need to find another strategy to induce that immune response to make the tumor go away. Sometimes there are biology and pathways which are unique to humans. They're not found in mouse, and so we need to develop drugs that are unique to humans. We don't have any other way of finding those drugs other than studying human tissue. And then there are other things that we do. We need to study tissue to try and figure out who's going to respond to a drug and who won't, because at the end of the day, in the future, we want to be able to give the right drug to the right person. We don't want to give an ineffective drug to somebody, but we want to give that same drug to somebody else because that other person might respond. And we want to be able to predict that. We want to make sure that the drugs that we're giving, as I said before, are at the right concentration, that we get enough of the drug into someone that it'll have the right effect. And that's measuring, that's called pharmacokinetics, or that drug is reaching the tissue, or that when it gets to the tissue, it does what it's supposed to be doing. And we get from all that information, we try and understand how the drug is working, why people are not responding. And again, we circle back, and that gives us new ideas about how to develop new drugs and new combinations. So I just want to touch briefly on how research is conducted. A lot of you will ask, well, what is a phase 1 trial? What's a phase 2? What's a phase 3? So when you have a new drug or a new combination, you don't really know the dose, the schedule. You don't know exactly how to give it. You don't know what toxicities it has. But this is a drug that you want to study because you have other information from mouse models or from studying human tissues that it might be effective. So the very first trial that we do with a new combination or a new drug is called a phase 1, which means we start at a low dose. And in groups of 3 to 6 people, we keep increasing the dose until we find the maximum tolerated dose. Now, usually we do phase 1 trials in people who have had other treatments and the treatments have not worked. And at that point, their best option are these new drugs because there isn't anything else approved for that disease. Now, once we know the right dose and the right schedule, and we know that it's well tolerated, we still need to know how active it is. And we don't get that information in the phase 1 trial. So then we take 30 patients who are very similar, who have the same disease, and then we offer them this drug. And again, we do it in an ethical way. We do it because it's probably the best option for them at that point in time. And then we determine how many of those people respond, and we use information from their tissue to try and figure out who did and who didn't respond. And then if we see enough activity, eventually we go to phase 3 trials. And phase 3 trials are randomized studies. A lot of you, when you hear about a randomized study, you say, oh no, I'm not going to get the drug, I'm going to get some sort of placebo. That's not really the way we do things in cancer. We're really testing the new drug against something that's already standard, or we're adding the new drug to a standard treatment and comparing it to the standard treatment to see whether that new drug actually adds to the activity of what we've already developed. So you can imagine that in the future, for example, if you take ipilimumab, nivolumab, which is a standard of care, in the future, we're going to try and add something to it. And first, we need to know the right dose, and then we're going to get some activity data. And then at the end of the day, we might have to compare that triple combination to the ipilimumab, nivolumab in a randomized trial. And the reason we do a randomized study is because we really don't know that that triple combination is going to be better than the doublet that was there as standard of care before. Some of you need to, it's very important to understand that the trials may or may not require that you have had prior treatment. Some trials are done in people who have had no prior treatment. Some trials are done in people who have received every possible treatment for their disease. And to go on a clinical trial, because these are very regulated activities, we have an eligibility criteria, and this is something that we agree on with the IRBs and the scientific review committees and institutional ethics committees and the FDA. And so sometimes we offer a clinical trial to somebody, but we have to tell them, I'm sorry, we offered you the study, but you can't go on the trial because the criteria don't allow you to go on. And these days, the regulatory bodies don't allow people who don't meet those specific criteria to go on a clinical trial. And that's done for safety reasons, because those criteria are designed to maximize safety for those drugs. So how do we pay for all this? Well, if you try and call Dr. Kluger at 10:30 or 11 at night, she's probably still up working in front of her computer writing a grant so she can fund her research. I unfortunately don't have to do that, but she does, and many of our other scientists have to do that in order to obtain funding. And Dr. Kluger and Dr. Bosenberg are directors of a very large grant that's given by the government called the SkinSpore, which funds very novel projects that help us develop new therapies for patients with cancer. Sometimes our clinical trials are funded by industry, so that means that industry pays us to collect the data. Sometimes we get large gifts from donors that help to support this research, and sometimes there are foundations and other types of grants that help us do either research in the lab or clinical trials that help to pay for what is a very expensive operation. So I'm just going to spend the next 2 or 3 minutes just giving you some examples of things that were done here or can be done. Some of it is our own work, some of it is work from other people. This is work done by Ruth Halaban, who was the director of the SPORE for 10 years before Dr. Kluger and Dr. Bosenberg took it over. And so this was a sequencing. We took lots of melanomas and we sequenced all the DNA, and we learned all of the mutations that are present in melanoma cells. Dr. Halaban discovered a type of melanoma, the one that has the NF1 mutations. And this kind of information, which tells us about the biology of the cells, it tells us the mutation, it tells us how the cells work, will help to develop new drugs. It'll help to develop new targets and new drugs so that people who no longer respond to the targeted therapies or the immune therapies can be offered therapies that have a chance of working for their disease. This drug, anti-Cichlid 15, this is another example of something that was discovered here at Yale. So we have a scientist, Dr. Liping Cheng. So all of you, many of you who have advanced disease have received anti-PD1 Opdivo or pembrolizumab. That pathway was actually it was defined to some degree by Dr. Chen. In fact, he was the first one to publish data showing that blocking that pathway could have anti-tumor activity. It's actually a crime that he didn't win the Nobel Prize for this. It was given to somebody else, but he certainly deserved it. So he's developed a screening methodology where he can actually find out what things help to activate your immune system, what things help to inhibit your immune system. And so you can imagine that we might want to give in the future drugs that help to activate your immune system, but the things that seem to work the best are things that are blocking your immune system, and we give drugs that block that blocking factor, so your immune system works better. And this is a drug that he found that seems to work by this pathway, the Cichlid 15, when it's expressed on your tumor cells or on cells that are inside your tumor, block T cell function. They block the cells that can kill the tumor. And so you can make an antibody that blocks this, and it helps the lymphocytes, the cells in your tumor, work better and help to attack your tumor. What was interesting about this is that this seems to work in the test tube where the anti-PD1 and anti-PD-L1 don't work. So this seems to be a pathway that's important in people who have a different way other than the PD1, PD-L1 pathway of blocking lymphocyte function. And we actually looked, this is gene expression data that Dr. Halaban generated, and we found out that there are some tumors that express PD-L1, but the ones that don't actually express high levels of Cichlid 15. And so in the future, this drug is already in clinical trials. We hope to take this drug and examine it in a subset of patients with melanoma, and we hope to see the activity, the similar activity that was seen in the test tube and in animal models. Here's another example, and I'll just cover this very briefly. This is work done by Dr. Bosenberg, who's the co-leader of the spore with Dr. Kluger, and by Dr. Keck, who's now at the Salk Institute. And they found that this pathway called CD40 is a very important pathway for the way some of these drugs work. And actually, it's a way of activating the immune system. And they showed that giving a drug that agonizes this pathway, an antibody against CD40, can cause very significant tumor regressions in mouse models, in some cases with a special group of cells called monocytes and macrophages. And without getting into the details, Dr. Kluger and Dr. Weiss wrote a clinical trial that combined this anti-CD40 drug, which was made by a small company, with the anti-PD1, the Opdivo, that many of you have received. And in the preliminary data that was generated here, we've seen incredible responses in 2 or 3 people who had previously progressed on the Opdivo, whose disease had not responded to the Opdivo. Now, this doesn't work in every patient, but this is the kind of preclinical work that leads to an idea, that leads to a clinical trial, that can cause a major change in treatment, especially for people who cannot respond to the anti-PD1 alone or the combination. And we have a couple of patients like this who've had wonderful responses. Now, this is early on. We'll have to see what happens. But this is an example of lab to clinic that was done here that may lead to a change in treatment. And I'm just going to show you here, this is this huge mesenteric mass that completely went away. This patient has done very well and is complete remission now, almost a year after starting this treatment. So I'm going to end here with the key research questions. The standard of care is giving Opdivo together with Yervoy, the anti-PD1, together with anti-CTLA-4. But when people don't respond, for the 50% of people that don't respond, we need to find new drugs. We need to bring that other 50% in and cure those other 50% that don't respond to those 2 drugs. And again, we need to find out at the very beginning those who will respond and those who won't respond, so we can give the curative therapy or those who are going to get cured, and the ones who would not get cured, we can predict and give them something else. We need to understand why people develop toxicity. Dr. Kluger is working on several different research avenues to try and understand who develops toxicity, why they develop toxicities, and better way to treat that toxicity or prevent that toxicity. There are people who won't respond to immunotherapy no matter what we do, and some of those can get these BRAF, these targeted therapies. But when people get targeted therapies, their disease responds very well. But at some point in many of those people, the disease starts to progress, and we need to find better treatments for those people, other molecular targets that can help their tumor. Dr. Kluger has started a major program in trying to figure out, as many of you know, we do a lot of brain MRIs because we're looking for disease that goes to the brain. In melanoma, this can be a major problem. We're trying to understand the biology of that, ways to prevent it and better ways to treat it. Dr. Kluger has a major program in place to try and understand the biology and develop better treatments. And in fact, one of the clinical trials that she developed, which is pembrolizumab in combination with bevacizumab, has shown very promising preliminary activity, and she showed you one of those slides. And finally, we're trying to develop better methods to detect the disease. So Dr. Weiss told you that we have monitoring that when people have a primary melanoma, some of those people will end up recurring in their disease, and we'd like to be able to find that recurrence with a blood test and not by doing scans. So finding methods to detect the disease early and treat it earlier may lead to better outcomes, and obviously less use of CAT scans. So those are the major areas of research that we're working on, and hopefully with time and a little bit of luck, we'll have solutions to some of these problems in the future. So thank you for your attention. I think at this point, I'll turn it back over to Dr. Kruger.